Today, I'm going to present on the effect of ApoE genotype on estrogen loss in EFAD mice. My name is Saharsh Narayanam, and my supervisor is Dr. Anna Valencia Olvera. Alzheimer's disease, or AD, is a neurodegenerative form of dementia that affects more than 10% of individuals over the age of 65. As of now, there is no known cure, but scientists have identified three universal biological variables, or UBVs, in age, genotype, and sex that greatly increase the risk for Alzheimer's. These UBVs, age, genotype, and sex, have been carefully described by many research studies in the past. Age is the greatest risk factor for AD, and AD risk increases significantly with older age. Genotype, ApoE4 allele is the greatest genetic risk factor for AD. The ApoE3 allele is the most common and tends to have a neutral effect, and ApoE2 is protective against AD, but is also very rare. Sex, females have a much higher risk of developing AD, and there's interactive effects in that females with the ApoE4 allele have an exacerbated risk for AD. AD pathology is diagnosed by the presence of both pathogenic amyloid beta and tau neurofibrillary tangles. And so the EFAD mouse model is able to take an ApoE transgene from humans and a 5X familial Alzheimer's disease mutation and combine them to create a new mouse model that overproduces amyloid beta 42, exhibits hyperphosphorylated tau, and expresses human ApoE isoforms. And the advantage of this is that it provides evidence of those genetic differences between ApoE2, E3, and E4 in terms of pathology. Lastly, estrogen tends to have neuroprotective effects against Alzheimer's, especially in preventing that amyloid beta buildup, and also may explain the difference between men and women. Because the women have a higher risk, many scientists think that the loss of the protective effect of estrogen after menopause greatly increases that risk for AD. And so in mouse studies, overectomy or OVX led to increased A beta levels, slower learning, and worse memory, suggesting that the depletion of estrogen leads to the exacerbation of AD pathology. Therefore, my hypothesis and study goals are to determine whether OVX will exacerbate the detrimental effects of the ApoE4 allele, resulting in a more severe increase in both soluble and insoluble A beta 42 in the E3-4 FAD and E4 FAD groups. My second hypothesis is that this effect will be heightened in the late procedure group compared to the early procedure group, which I'll explain later, due to AD pathology being highly dependent on age. And so to explain that early and late groups, there's an early OVX and a late OVX group in which the procedure, OVX or sham, is done at either 4 months or 8 months, and the groups are sacrificed at 8 months or 12 months, giving them different age ranges. And so the results of my study indicated that A beta 42 formic acid fraction, A beta deposition, and ApoE formic acid fraction was lowered in E3 FAD OVX while increased in E4 FAD OVX compared to sham, but this trend was only in the early groups. And so these three data points indicate three things: the impact of age on pathology, in which the age at which the procedure, both sham and OVX, was conducted, significantly changed the levels of insoluble ApoE and insoluble A beta 42, corroborating the fact that AD pathology worsens with age, especially amyloid deposition. Once again, proving that age is the single largest risk factor for AD. Furthermore, ApoE genotype greatly impacted pathology as well, in which we saw that the ApoE4 allele generally increased insoluble A beta levels and A beta deposition levels in both early and late groups compared to ApoE3. And we think that this increased deposition can be attributed to the lower amyloid clearance rates by those ApoE4 bound lipoproteins. And lastly, the impact of ApoE genotype on OVX was significant, in that only in the early treatment groups, A beta deposition and insoluble A beta data indicated that the decreased A beta levels in E3 FAD mice and increase in E4 FAD mice. And so, but the E3-4 FAD mice had no difference between sham procedure and OVX procedure. And so it's clear that the ApoE4 allele interacts with OVX to present worsened AD pathology. However, one caveat is that given past literature, one would expect that OVX would worsen pathology in E3 fat mice as well. However, a potential reason for this in our results is that the ApoE3 allele seems to be able to mitigate the detrimental effects of estrogen loss via OVX. And so perhaps the mice are able to adapt to that loss of estrogen and kick ApoE3 associated amyloid clearance into overdrive in order to decrease the amount of amyloid beta. However, one thing I noticed is that this trend was only in the early group. And so one possible explanation is that once we get to the late group, A beta levels may plateau at a certain age, leading to less observable differences between the genotypic groups. And so we can conclude from my results that age, a genotype, and sex all contributed to greatly increase Alzheimer's risk, and that the older mice had worse pathology, the ApoE4 allele had worse pathology, and OVX, when interacting with the E4 Fad mice, show that loss of estrogen interacts with the ApoE4 allele to exacerbate AD risk. And so future directions for my study include investigating whether estrogen treatment and its alternatives can alleviate this effect of estrogen loss in E4 fat mice and decrease that AD pathology that we don't want to see. Furthermore, we can investigate the mechanism behind that decrease of amyloid that was only in the E3 fat mice following OVX, and this could potentially have therapeutic implications. Thank you all for listening.